What if a nation of 5.5 million people holds genetic secrets that could completely rewrite human history? That their DNA contains clues to one of the most extraordinary migration stories ever discovered, a journey so incredible that scientists refused to believe it for decades. Finland isn't just another European country. It's a living genetic time capsule that preserves evidence of an epic 4,000-mile odyssey from the frozen wilderness of Siberia to the shores of the Baltic Sea. Only 10% of Finnish DNA comes from this distant homeland. Yet somehow these ancient Siberian travelers managed to completely transform the genetic and linguistic landscape of Northern Europe. How is that even possible? The Finnish people represent the most confounding genetic puzzle in Europe, and recent discoveries have made this mystery even more incredible. While every neighbouring population speaks Indo-European languages and carries the standard genetic signatures of ancient European farmers and steppe herders, the Finns are genetic outliers who shouldn't exist according to traditional models. Their genes whisper secrets of distant Siberian origins that span thousands of miles. Their language connects them to Hungarian and Estonian through linguistic threads that stretch across continents. But most shocking of all, their DNA reveals population bottlenecks so severe they created an entirely unique category of genetic diseases found nowhere else on Earth. We're talking about a people whose genetic isolation was so extreme it literally rewrote the rules of human genetics. But everything scientists thought they knew about Finnish origins just got turned upside down. In 2024, researchers published a groundbreaking study in Nature that analysed ancient DNA from 180 newly sequenced Siberians, and what they found defied every expectation. The ancestral homeland of Uralic speakers, the linguistic family that includes Finnish, wasn't in the Ural Mountains where linguists had placed it for over a century. Instead, it was located thousands of kilometres further east, in the Yakutia region of northeastern Siberia, closer to Alaska than to Finland itself. Think about that for a moment. The language you hear spoken in Helsinki today originated in a frozen wilderness that's practically touching the Arctic Ocean. Exactly how these ancient people managed to carry their culture across the breadth of the entire Eurasian continent. One can only imagine Finland 13,000 years ago, one of the most hostile environments humans had ever attempted to inhabit. The entire landscape was buried under ice sheets over a kilometre thick. Imagine the Empire State Building, then stack 3 more on top of it, and you'll begin to understand the scale of ice that covered this land. As the last ice age finally began to wane, retreating glaciers revealed a pristine world of exposed bedrock, emerging islands, and gradually warming tundra. This was humanity's ultimate survival challenge, and the people brave enough to take it on were about to establish genetic lineages that would influence European history for the next 13 millennia. The first humans to set foot in what would become Finland were Eastern European hunter-gatherers, and they carried something precious that would prove absolutely crucial to the genetic puzzle we're unraveling today. These pioneers belonged primarily to mitochondrial DNA haplogroup U5b, one of Europe's most ancient maternal lineages with roots stretching back to the Paleolithic era. But here's where it gets really interesting. The Finnish variant showed specific subclades U5b1 and U5b2 that linked them directly to Eastern European forager populations around the Don River basin, not the Western European Cro-Magnon peoples who were simultaneously colonizing other parts of Europe. This Eastern connection was the first genetic thread that would eventually link Finland to the vast Siberian wilderness. But these early settlers had no idea they were laying the foundation for one of history's most remarkable demographic stories. What's absolutely fascinating is that modern Finns still carry a massive 25% of this ancient U5b ancestry in their maternal lineages. This isn't just genetic curiosity. This represents one of the most successful examples of population continuity in European history. These ancient hunter-gatherers didn't just survive in the harsh northern environment. They thrived so successfully that their genetic signatures became permanently embedded in the Finnish gene pool. Archaeological evidence from sites like Ristola in Lahti shows they possessed incredibly sophisticated adaptation strategies, crafting microliths, tiny, precisely shaped stone tools that could be hafted onto spears and arrows for hunting massive reindeer and elk. But they were also establishing something even more important, the demographic foundation that would allow Finland to absorb later migrations while maintaining genetic continuity with Europe's earliest populations. As Finland's landscape stabilized during the warming Holocene, a remarkable cultural phenomenon emerged that would fundamentally alter the genetic trajectory of Northern Europe: the comb ceramic culture. Named for their distinctive pottery decorated with intricate comb-like impressions, these people brought far more than new artistic styles to Finland. They carried genetic signatures that connected Finland directly to Siberian populations. And they maintained these connections for nearly 3,000 years, from around 4,200 BC to 2000 BC. Despite their name suggesting farming connections, the comb ceramic people remained dedicated hunter-gatherers, who successfully resisted the agricultural transitions that were sweeping across Europe. This resistance to farming wasn't just cultural stubbornness. It was a demographic strategy that would prove essential for preserving the ancient genetic lineages that make Finnish genetics so unique today. The comb ceramic people's sophisticated pottery served specialized functions in processing and storing resources from Finland's abundant aquatic environments, and recent analysis of pottery lipid residues reveals they were exploiting incredibly diverse food resources without requiring agricultural supplementation. Sites like Kirikki north of Oulu revealed year-round habitation and extensive trade networks that brought flint from Scandinavia, amber from the Baltic, and slate from distant regions. These weren't isolated primitives, they were running sophisticated trade networks across vast distances while maintaining the genetic continuity that would prove crucial when the most transformative migration in Finnish history finally arrived. The genetic continuity maintained by comb ceramic populations created the perfect demographic foundation for what was coming next. Around 2800 BC, Finland's genetic isolation was shattered by the arrival of corded Ware peoples. These weren't peaceful migrants. They were part of massive population movements associated with Indo-European expansion that were reshaping European genetics from the Atlantic to the Ural Mountains. The corded Ware culture was established primarily by people from the Yamna culture, carrying 75% of their autosomal DNA, along with Y chromosome haplogroups, mainly R1a, specifically the R1a Z645 subclade, that would become a defining genetic marker of Indo-European expansion. The corded Ware impact was geographically restricted to southwestern Finland, creating distinct cultural and genetic zones that would prove essential for understanding how later Siberian influences would penetrate different regions through completely different demographic mechanisms. The period featured 3 distinct archaeological zones in southern and central Finland: the corded Ware culture, the Giant's Church culture, and the Finnish Lake District, with obvious cultural boundaries but also complex interactions between zones. The integration of corded Ware elements with existing comb ceramic traditions created hybrid cultural forms, ultimately giving rise to the Kiukainen culture that existed between 2300 BC and 1500 BC. This cultural synthesis reflected complex demographic processes rather than simple population replacement, demonstrating that indigenous populations maintained significant influence even as they adopted new technologies and cultural practices. Crisis struck around 2500 BC when massive population decline and social organization collapse appeared across the region, possibly linked to Yersinia pestis epidemics that devastated late Neolithic populations throughout the eastern Baltic. These catastrophic events amplified the genetic impact of surviving populations, contributing to founder effects that would characterize Finnish genetic structure, while simultaneously creating demographic opportunities for new populations to establish themselves in previously occupied territories. Now we reach the most extraordinary chapter in Finnish genetic history, the introduction of Uralic languages and the genetic signatures that accompanied them. Recent research has completely revolutionized our understanding of this process, revealing origins so far east they challenge everything we thought we knew about ancient human mobility. New research published in Nature identifies the prehistoric Uralic homeland as existing about 4,500 years ago in northeastern Siberia within Yakutia, geographically closer to Alaska than to Finland. This discovery represents one of the most remarkable revelations in the study of language and population movement, demonstrating that human migrations could connect regions separated by distances that seemed impossible until very recently. The genetic signature accompanying Uralic expansion into Finland is dominated by Y chromosome haplogroup N1c, which has become the defining paternal lineage of Finnish populations and provides the clearest genetic evidence for Siberian origins that we could possibly ask for. The Bolshoi population from the Kola Peninsula, dating to around 3,500 years ago, carried Y chromosomal haplotype N1C1A1A1 N-L392, representing the earliest known occurrence of this Siberian genetic marker in Fennoscandia. This genetic signature serves as a direct tracer for the westward movement of Uralic-speaking populations from their distant Siberian homeland, providing concrete DNA evidence for one of the most extraordinary migration events in human history. But here's what makes this migration even more incredible. It was facilitated by technological innovations that gave these Siberian populations competitive advantages across the vast expanses of northern Eurasia. The Uralic expansion coincided with the Seima-Turbino Phenomenon, the sudden appearance around 4,000 years ago of technologically advanced bronze casting methods across northern Eurasia. These technological advantages provided Uralic speakers with superior capabilities in resource extraction and long-distance trade that facilitated their demographic expansion across territories spanning thousands of miles. The genetic composition of these early Uralic speakers reveals the complexity of ancient population movements that would create modern Finnish genetics. Ancient individuals from this period carried distinctly Siberian mitochondrial DNA haplogroups, Z1, C4, and D4, which are still common in modern Siberian populations today. These genetic markers provide direct evidence of Siberian origins, while demonstrating successful integration with pre-existing Finnish genetic structure, creating the unique admixture patterns that characterize modern Finnish populations and distinguish them from every other European group. Recent linguistic evidence supports this remarkable migration while revealing how languages can spread through demographic processes that leave surprisingly subtle genetic signatures. Revised chronology places the dating of Proto-Sami, Proto-Finnic, and Proto-Uralic as clearly younger than previously thought, contradicting continuity theories and supporting recent arrival scenarios that emphasize demographic movement rather than purely cultural transmission. But this creates the most fascinating puzzle of all. How did relatively small populations carrying only 10% genetic ancestry manage to achieve complete linguistic dominance across such vast territories? The answer lies in the unique demographic integration that occurred when Uralic speakers reached Finland. Unlike the geographically restricted impact of corded Ware expansion, Uralic influence spread throughout Finland, creating the genetic foundation for modern Finnish populations, while maintaining sufficient demographic continuity to preserve earlier genetic patterns in specific lineages and regions. This pattern suggests that Uralic speakers either arrived in larger numbers than archaeological evidence indicates, or possessed cultural and technological advantages that enabled rapid demographic success despite their distant origins and relatively small genetic contribution. Recent 2025 research analyzing approximately 1,800 Finnish Y chromosomes has revealed even more complexity in this migration that challenges our understanding of ancient population movements. The study found evidence that haplogroup N carriers migrated to Finland via multiple routes, not just the expected eastern pathway, with one N subclade common in southwest Finland, arriving through the Baltic region from the south. Finnish Bronze Age marked a period of genetic consolidation and increasing isolation that would prove absolutely crucial for shaping the unique characteristics that distinguish modern Finnish populations from every other group in Europe. Beginning after 1500 BC, coastal regions participated in Nordic bronze culture, while inland regions received influences from bronze-using cultures of northern and eastern Russia, reflecting Finland's geographic position at the intersection of multiple cultural spheres, while maintaining distinctive demographic characteristics that would have profound consequences for population genetic structure. The genetic isolation that characterized Bronze Age Finland created the demographic conditions responsible for one of the most unique aspects of Finnish genetics: the Finnish disease heritage. Population bottlenecks among ancestors of modern Finns occurred about 4,000 years ago, resulting in exceptionally low diversity in the Y chromosome that reflects the survival of just 2 ancestral male lineages. These demographic constraints created optimal conditions for genetic drift and founder effects that would ultimately generate the collection of genetic diseases occurring at unusually high frequencies in Finnish populations. Diseases so specific to Finnish genetics that they formed their own medical category. The Finnish Iron Age witnessed the crystallization of Proto-Finnic genetic and cultural identity through nearly 2 millennia of demographic consolidation that would establish the population structure persisting into historical times. Lasting from 500 BC until 1300 AD and divided into 6 sub-periods with distinct regional cultures, Finnish proper, Tavastian, and Karelian. This extended period allowed for complete development of genetic patterns characterizing modern Finnish populations, while maintaining connections to broader Northern European demographic history. Iron Age genetic evidence reveals the extreme demographic processes that shaped proto-Finnic populations, while providing crucial insights into the relationship between Finns and their linguistic relatives. Iron Age Fennoscandian sites showed strong genetic drift, with high levels of linkage disequilibrium and low diversity in uniparental markers that explain why some ancient mtDNA lineages completely vanished from present-day populations. Iron Age sites displayed relatively high frequencies of U4 subhaplogroups, U4A-U4B and U4D, which are rare in contemporary Finns and completely absent from modern Sami, but exist in high frequencies in the Volga-Ural region. Genetic relationship between Sami and Finnic populations represents one of the most fascinating aspects of Northern European population history, revealing both shared origins and distinct evolutionary trajectories that illuminate the complex demographic processes shaping the region, while providing crucial insights into Siberian origins, linking both populations. Sami languages split from their common ancestor about 3,300 years ago, with autosomal genetic analyses finding that Sami people cluster in a distinct position, shifted away from other Europeans, carrying significant proportions of genome originating from Neo-Siberian source populations best represented by modern Nganasan people. Classification of Sami mtDNA lineages revealed that the majority cluster in a specific subset of the European mtDNA pool, with haplogroups V and U5b dominating and accounting for about 89% of total lineages, giving Sami regions the highest levels of these haplogroups found anywhere in the world. Y chromosome patterns in Sami populations display complex geographic variation, reflecting multiple waves of demographic influence and historical migration patterns that reveal unexpected connections to broader Eurasian population history. Haplogroup R1a in Sami occurs mostly in Swedish Sami and Kola Sami populations, with low levels among Finnish Sami, suggesting that both N1c and R1a reached Fennoscandia from Eastern Europe, where these haplogroups exist in high frequencies today. This geographic variation within Sami populations reflects differential demographic contact with neighbouring populations and historical migration patterns, while preserving evidence for ancient genetic connections linking Northern European populations to their distant relatives. Archaeological evidence suggests that ancestors of Sami populations occupied much larger territories during prehistoric periods than their current distribution indicates, revealing how historical processes can dramatically alter population distributions while preserving genetic evidence for ancient demographic patterns. The medieval period brought unprecedented demographic changes to Finland through Swedish expansion, crusades, and systematic trade networks that would alter Finnish genetic structure in regionally specific ways, while simultaneously preserving ancient genetic heritage established over millennia of demographic history. Written records become more common due to northern crusades led by the Catholic Church in the 12th and 13th centuries, documenting historical processes that introduced new genetic elements while preserving demographic patterns that had developed during preceding millennia. Swedish colonization had profound genetic consequences, particularly along Finland's southern and western coasts, creating the modern pattern of genetic differentiation that continues to characterize Finnish populations today. Large YSTR differences between western and eastern parts of Finland reflect regionally restricted gene flow extending to southwestern and northern Finland, while eastern regions retained more Finno-Ugric genetic composition originating from regions east of Finland. This pattern created the enduring genetic dichotomy characterizing modern Finnish populations. The introduction of Scandinavian genetic elements occurred through organized settlement, military campaigns, and trade relationships. But the geographic specificity of this influence reveals how historical processes create lasting patterns in population genetic structure. Scandinavian influence appears as an additional genetic element in Western regions, whereas the Finno-Ugric genetic component remains more prominent in eastern Finland, with Scandinavian haplogroup high occurring at frequencies exceeding 30% only in western Finland. The genetic origins of the Finnish people represent one of the most remarkable stories of human demographic history, spanning 11,000 years of migration, adaptation, and survival in Northern Europe's challenging environments, while revealing connections that span continents and provide crucial insights into how languages and populations spread across our planet. From the first post-glacial hunter-gatherers, carrying ancient European lineages to the complex admixture events that introduced Siberian ancestry through Uralic-speaking populations, Finnish genetic history illuminates the intricate processes that shaped human populations throughout northern Eurasia. The Finnish genetic story continues evolving as new research techniques reveal ever more complex patterns of ancient population movement, challenging our assumptions about the past, while providing crucial insights into the demographic processes that shape the modern world and continue to influence human genetics today.